Okay, so our third family of macromolecules are proteins, and they have the elements carbon, hydrogen, oxygen, and now they have nitrogen. They have also, in some cases, they'll have sulfur, but not all amino acids have sulfur, but all amino acids do have nitrogen. They all have carbon, hydrogen, and oxygen. So that's found in all amino acids, but the sulfur is found in some amino acids, in particular like cysteine, and that's going to be something we're going to talk about with a biochemical test that releases sulfur when it breaks down, and so that is going to be important later on. The monomer, the bond, before we start with that, the bond for proteins is called a peptide bond, and it's formed between amino acids to make a dipeptide. Some examples of amino acids are glycine, tryptophan, and cysteine. So a basic structure, we'll look at that on the PowerPoint here in just a minute, has an amine group and an acid group. It has a carboxyl. So it has those two parts, the amine group, and the acid is the COOH, the carboxyl, and the amine group is NH2. So you have an amine group on one end and a carboxyl on the other end on all amino acids, and they're kind of lined up in that order. So you have a big long chain. All of the amines are pointing in the same direction because the peptide bond is formed between the amine group of one and the carboxyl of another. So dipeptides, meaning it has two subunits. Polypeptide, meaning that you have three of the subunits. And some examples of the we're not going to worry about examples of dipeptides, but the polymers, the polypeptides like enzymes are, and this is not an exhaustive list, but an enzyme is a really important type of protein. It's a catalyst. It speeds up chemical reactions. Antibodies are important in immunity. The cytoskeleton is one of the polymers. It's going to give structure to the cell, and cytoskeleton is made of various different kinds of proteins like tubulin or myosin or actin. Those are all cytoskeletal structures, and they're made of protein. And most hormones are made of protein. Now, not all of them, but they're there for, again, regulation. There are steroids that are hormones that are lipids, but all the other proteins or all the other hormones are generally hormones or proteins. So proteins are the shapers of life. They, again, their particular structure determines their behavior, the unique qualities of organisms, and so that is one of the predominant macromolecules in cells. The amino acids are the building blocks, the monomers, and there's 20 different amino acids that are naturally occurring, and they're linked by peptide bonds. So here are the 20 amino acids by their abbreviations. So they have the 3-letter abbreviations, and some of them, just to save room in the computers, they have given them a letter of the alphabet since there's 20 of them. Here you can see they have a basic structure with an alpha carbon in the center, and then a carboxyl group on one end and an amine group on the other end. So all of them have that basic structure of the amine group on one end and the carboxyl on the other end, and then they're both attached to this alpha carbon, which has a hydrogen attached to it, and then this is the rest of the molecule. And whatever the rest of the molecule may be, that's going to determine the unique characteristics, the unique shape of the molecule. So the peptide, a peptide, when we say peptide, we're talking about a short chain of amino acids. A polypeptide generally has more than 20. Actually, I would say more like, well, a short polypeptide chain, a small polypeptide would be about 20. And then larger polypeptides, what we would consider a protein, would be probably 100 amino acids. So not all polypeptides are large enough to be considered proteins. And the protein structure, when you just have the number, the type, the number, and the order of the amino acids, that's called the primary structure. When you have the hydrogen bonds being formed and functional groups on the outer surface of the molecule interacting, then you have the alpha helix and the beta pleated sheet. And so that's a secondary structure. So you're just a list of amino acids, that's a primary amino acid structure, or a primary structure of proteins. When you get the hydrogen bonds forming, then that's a pleated sheet or an alpha helix or a random coil. Those are all secondary structures. So the tertiary structure is the folded, complete, the folded polypeptide chain, and it's just one polypeptide. But here, when you have two or more polypeptides, this is called the quaternary structure. So this is the tertiary structure when you just have one polypeptide chain. When you have more than one, and that's really the only difference. They have, within them, they have some alpha, we have some beta, or random coils, maybe some beta sheets in here. And so those combined together make this globular structure that is the tertiary structure of the protein. So some examples again, the enzymes are catalysts, protein catalysts. They speed up the reactions within the cell, and they're super important because pretty much any kind of reaction within the cell, it's going to need an enzyme to make that happen at the speed that it needs to happen within the cell to maintain life. And antibodies are necessary for, they attach to the surface of bacteria and viruses and other microorganisms, and that causes them to be inactivated, neutralized, so on, and it attracts other parts of the immune system. So they have lots of different functions, and we'll talk about that when we get into the immunity section in Unit 5. But antibodies are going to be involved in immunity. And when we talk about the shape of the protein, we say if it's in its native shape, that's the functional three-dimensional form, the globular three-dimensional form of the protein. And if you heat it or change the pH, that will disrupt the bonds, the hydrogen bonds, and that will cause it to break apart, and the hydrogen bonds break. Now the covalent bonds are still there, but the hydrogen bonds break, and so then they lose their shape. So it's a disruption of the native shape, native state of the protein when you denature. So when you think about like Jell-O, if you leave it out at room temperature for a long enough period of time, the heat is enough to break the gelatin bonds. Gelatin is actually not a sugar. Sugar is combined with the gelatin to make it sweet, but gelatin is the protein that actually gives it its shape. And if you leave it out long enough, then it kind of melts and is, melting is not a very good word for it, but it denatures and it becomes liquid.